Okay, you haven't had a screencast from me in a while, so in this screencast we are going to talk about transport in and out of cells. So when we're referring to the membrane, really we're talking about the membrane of a cell. And we already briefly talked about hypotonic, hypertonic, isotonic solutions, and we talked about what can happen when things come in and out of a cell. But just to get this really into your head, let's go through it a couple more times. So to put things into context, remember that all living things, whether that is a tree or a single-celled living thing like this euglena that we have here, we'll look at them with a microscope in a bit, or a human being, they're all going to need to release things, whether that's, sweat from performing, whether it's another form of waste. There we have all of those pink arrows showing the waste, all of the things that are being released. But they also need to be able to bring things in order to survive and reproduce. So let's draw some green arrows coming into these living things. So all living things constantly need to interact with their environment. And remember that all of these things are made of cells. Trees, obviously lots of cells. Something like a euglena, it's only one cell. A human, made up of lots of cells. But no matter what, they all will need to interact with their environment. So inputs and outputs are critical for all living things. And when things are moved in and out of a cell, when we're talking about this in technical terms, we call that membrane transport. And this is something that has to be controlled. And Mr. Chang has a good analogy that he likes to use. He likes to think of it like an airplane. When you get on an airplane or you get off an airplane, it will happen in a controlled way. You can't bring anything you want onto a plane. You have to go in a line and in an orderly fashion. And so that is similar to what's happening in cells. Right, to get on a plane, you know you need a boarding pass, you need to have luggage that's a certain size, or else it has to go and be checked. You know that the luggage that you do check, it has to, follow certain procedures and only contain certain things. And if there's something in your luggage that you're not supposed to have, then they're not going to let that on the plane. So there's a control that needs to happen when getting into and out of an airplane, which is similar to cells and having things enter and leave cells. But before we do that, let's look at the plasma membrane. We've talked a little bit about its makeup, but let's review. The plasma membrane is what we call a lipid bilayer, or a phospholipid bilayer, because the layer is made up of a phosphate head, which then has two fatty acid tails that come off of it. So this whole structure here, right, what we call a phospholipid, they make up the cell membrane or the plasma membrane, and often they'll make up any other membrane that you have for an organelle. But let's show what that would look like to have a whole barrier made up of these. So there's one layer, but it's a double layer, and the heads are going to be facing opposite directions, and the tails will face each other. That's because of their affinity or attraction to water. The phosphate heads are going to be hydrophilic, and the tails are going to be hydrophobic. So let's say that this is outside of the cell, and then in here is inside of the cell. We know that there will be water outside of the cell, most likely in its environment, and you'll also find that water inside the cell, which is why those tails are facing away from those areas. This image is a little better, but you can see again, each of those yellow heads are the phosphates, and then the little brown sticks that look like they're coming out are the tails. But also in a phospholipid bilayer, in the cell membrane, we don't just have those lipids or fats, but we also have these proteins. So you can see them shown in a pinkish color. And they typically are used as channels to allow certain things in and not allows other things. So some things may be able to pass really easily through this lipid bilayer. They can move apart if it's really small, but sometimes you're going to need channels to allow things to come in and out. And you'll also find carbohydrates that are occasionally attached to the cell membrane as well. So let's get back to our metaphor. And again, we're thinking like of cells as being like airplanes, and then the plasma membrane of a cell, that being like sort of the ticket agent or baggage handler at an airport, which will tell us what can go on and what can go off, or who can go in or out of a plane. So let's start with some basics about how this actually happens in a cell. The first thing that we should review is this idea of something called diffusion. Diffusion is the movement of materials from an area of high concentration to an area of low concentration. And when we're talking about cells, it's important to remember this word concentration, because you probably have heard of something diffusing before, but it's important to remember in terms of the amount that exists, or the concentration, and it going from an area of high to an area of low. But let's quickly review what concentration is. If someone opens this bottle of perfume, there's going to be obviously a high concentration of perfume right there, but when it's opened, then let's say there's a person who's standing over here, far away. Right now, initially, they can't smell the perfume because there's currently a low concentration of those perfume molecules near the individual. But eventually, those molecules will start to spread out if a person opens it or sprays it, until eventually they will head over towards where there's a low concentration of those molecules. And that is because the molecules will move and diffuse from an area of high concentration to an area of low concentration. And a key thing to remember is that there is no energy needed for this process. Okay, this just has to do with the movement of molecules. It's not requiring energy. It's based entirely on molecular motion, where things that are highly concentrated together are going to bounce around and hit each other and sort of bounce away and move away and spread out evenly throughout the entire area. When diffusion's happening, we are moving down what is called a concentration gradient. So here's a concentration gradient that I've drawn. We can see there's high concentration here, and it gets lower until you get over here where you really only have two of those dots. So when we say that something is moving down its concentration gradient, we move that it's moving from an area of high to low. And hopefully from chemistry you remember that when we're talking about concentration, we put these little brackets around things. So this means high concentration, and that means low concentration. Okay, moving on. You also need to know about something called solute. Solute. And you also need to remember about solvent. If you're taking chemistry, these words should sound familiar to you as well. Remember that the solvent is what's doing the dissolving. And in biology, often we are referring to water as the solvent. And the solute is the thing that actually dissolves into the solvent. So in this example with an Alka-Seltzer dissolving in water, we know that the water that we have down here, this will be our solvent, and in the water we are dissolving these Alka-Seltzer tablets, so that will be the solute. Okay, the next thing we need to keep in mind is something called permeability. So this isn't something that's super complicated, but here's a good way to think about it. Think about it as a metaphor for clothing. So let's say that it is raining outside. That's not so great, but you want to keep yourself from getting wet. If you wear this really beautiful unicorn sweatshirt here, it's not going to do a great job of keeping you dry. So this is something that we call permeable. It's a very permeable sweatshirt. Even though you can't really see it with a naked eye, it has lots of holes in it. So if you're going to wear it and you're outside and it's raining, you will get wet. But then instead, if you are wearing this rain slicker, especially if you have that really cool hat on with those ears, these are not permeable at all. They keep you pretty dry, but that means that no rain is able to get through that material and onto your body. But if you were to wear that raincoat and that hat while you're running around and exercising, you would get really sweaty. And the reason is because that sweat can't leave your body, it can't wick away, and it's keeping all of that material inside. So that's an issue when you have a non-permeable substance. It traps all of that perspiration next to your body. We sometimes also call that impermeable. But cell membranes are going to be kind of in between these two things. They're kind of like a Gore-Tex jacket, actually. And what's great about these jackets is they allow perspiration, your sweat, to actually escape, but they won't let drops of water in and make you wet from the rain. So this Gore-Tex jacket, it selects what can go in and what can come out of the material. So we'd call this Gore-Tex jacket and also your cell membranes selectively permeable. Some people may call this semi-permeable instead, but we're not going to use that word. You may hear other students that use it instead, or textbooks, but semi-permeable means it kind of is, but selectively means it's choosing what can and cannot come in and out. So we've covered a bunch of basics. We started talking about diffusion. We talked about how that is related to a concentration gradient. We talked about solute and the difference between that and a solvent. And then we finished up by talking about permeability. So those are the basic things that you need to keep in mind as we go on to talk about equilibrium and osmosis and different types of solutions that can exist in the next screencast.